Hi everyone, my name is Alessandro Rossetta. I am the CEO and founder of FlameLabs, and FlameLabs develops devices and software for fluorescence lifetime analysis. Today I am here at Fondazione Policlinico Campus Biomedico because FlameLabs is also the coordinator of a EIC Pathfinder project named LaserBlood, which has with the ultimate goal of developing a technology for the early detection of pancreatic cancer.
Hi, I am Marco Casadei and I am a research scientist at FlameLabs. We are here today in Campus Biomedico Rome for the consortium meeting of the LaserBlood project. The LaserBlood project is founded under the EIC Pathfinder framework, which is a framework of funding inside the European Innovation Council. It is the first step that an innovation can take in the funding of this type of the research fundings, and it's the one where the technology gets developed. So we are basically starting from preliminary results and scientific publications, and the goal of the project and in general of EIC Pathfinder projects is to take what is scientific evidence to technological evidence. So to reach TRL4, technological readiness level 4 by the end of the project, which is of course before actual commercialization, but it's already in let's say proved in a laboratory environment.
Pancreatic cancer is a very challenging disease to be detected in its early phase. Damiano Caputo, surgeon at Fondazione Policlinico Universitario Campus Biomedico. Pancreatic cancer is often asymptomatic at the beginning, and when symptoms like pain or jaundice are evident, the disease is already in a late stage. To date, we do not have any instruments or marker to use in clinical practice to achieve an early detection of this cancer or to be used on a screening on a large population. There is an urgent need to have new tools and instruments to achieve these goals, because the detection in the earlier phase of the disease can lead to better results after the cure of this dreadful disease. I am a clinical partner of the project LaserBlood. We collect plasma samples from pancreatic cancer patients and IPMN patients, and these samples will be then given to the other partners of the consortium.
We collected the blood sample from PDAC patients affected by pancreatic ductal adenocarcinoma. We achieved the plasma sample and sent these samples to the nano delivery lab of Sapienza University of Rome to do the analysis of the corona proteica.
In Sapienza, we have been exploring the interactions between nanoparticles and biological fluids for the last 15 years or so. When you incubate nanoparticles into biological fluids, such as human plasma, urine, saliva, and so on, these nanomaterials are coated by protein corona that contains a signature of a certain disease. In the LaserBlood project, we are going to develop a nanoparticle-enabled biophotonic technology for the early detection of pancreatic cancer. We are supposed to receive plasma samples, and we will expose a library of nanoparticles to plasma samples in order to generate these protein coronas. And we will then deliver them to FLIM labs, so we are supposed to perform FLIM experiments in order to look for and hopefully find a biophotonic signature of the disease.
As a researcher of the Nano Delivery Lab of Sapienza, led by Professor Giulio Caracciolo and Professor Daniela Pozzi, I'm involved in the experimental section. What we do is to incubate nanomaterials with biological fluids such as plasma from donors. In this project, we used also engineered murine plasma, and the aim is to study the very early stage of pancreatic cancer disease in a mouse engineered for tumor progression. So this is a very impressive investigation, because it's very difficult to study the early stage of this tumor in a human subject, because it's a tumor that is difficult to detect at this early stage. So in this project, we try to detect some protein biomarker, but also other biomolecules in the plasma derived from first murine models and then human subjects. And another strength of this project is also to study high-risk population that develop pancreatic cancer, such as IPMN subjects.
This project will overcome the current gaps in the PDAC detection, the early detection of these malignancies, that it's silent in its first part, so it's really hard to detect. And it will be not only important for the detection, but also for the treatment, because there will be more option for the treatment of this pathology if detected before.
We are all excited for this new project, which may have a great impact on the pancreatic cancer diagnosis. This malignancy is difficult to detect and it has a high mortality.
We actually developed during the past years several animal models for different tumor disease. My name is Giulia Piaggio and I am a group leader in the Cancer Institute of Rome, Regina Elena. And in focus on the LaserBlood project, I would like to say some words about a model that in which is very useful to follow pancreatic cancer. Indeed, several years ago we developed a mouse model in which all proliferating cells are bioluminescent, and trace for proliferation, bioluminescence trace for proliferation. In this way we can follow very early step of transformation of pancreatic cancer, and this model is actually very useful for the LaserBlood project. Indeed, we are collecting several plasma samples at different stages of pancreatic cancer development. We are fascinated by the technology that Flim Labs and Sapienza University put together, because we are sure that using this preclinical model, very homogeneous preclinical model, will allow us, together with the technology, to develop a tool really useful to be employed in clinical settings.
In the LaserBlood project, I work on the preclinical model of pancreatic cancer. To date, this preclinical phase is essential and fundamental for the transition to the medtech and biotech development stages. It provides the necessary foundation for any further technological applications.
Hi, I'm Christian Pilarski. I'm from the University Hospital of Erlangen, and we are providing samples from pancreatic cancer patients for LaserBlood. What we do if we have patients in our hospital, we go to the patients, ask them if they want to participate in a sample collection for different studies, and then we process the samples based on the SOP which was provided, and then send the samples to the Sapienza group, which will then do the analysis. We also take for these samples clinical data for the association of the marker with potential clinical disease.
My name is Emilio Llanos. I work as a Quality and Regulatory Affairs consultant at Complear, a Portuguese company that focuses on regulatory and quality affairs for medical device software solutions, but also for medical devices, traditional ones, and in vitro diagnostics devices has the LaserBlood. About regulations and quality, it's really important to focus on the requirements that we need to cover for the medical device industry. The medical device industry is a highly regulated environment that you should take into account to move forward into the project management and specifically in the design and the development phase. You have to cover that requirements early in the project. When you face a project like LaserBlood, you have to take into account that you have to cover all the requirements from the beginning in terms of regulatory and quality, but specifically with the regulatory. Because, for example, what kind of definition that you give about your medical device have to be clear, concise, and structured. And if you don't have it, you will have many obstacles in the future to go to the market and to develop a proper strategy.